Hello, everyone. It's a pleasure to be here. I'm Jay Hoying, Chief Scientist at Advanced Solutions Life Sciences, and I want to talk to you today about our work in the area of developing in vitro tissue models that are vascularized and made entirely from human constituents. So here at Advanced Solutions, we focus on really two types of in vitro models. One is modeling tissues derived from cell systems and combinations that give us the outcome that we want, as well as combining then these tissue systems into more complex, more organ-like, or chaining them together into looking at physiology. And as a reminder, remember that organs such as the heart shown here on the left are made from tissues, which there may be multiple tissue types contributing to that organ, but each tissue then is made up of a variety of constituents, including different cell types, whether it's in the case of the myocardium here, cardiomyocytes, cardiac fibroblasts, the microvasculature, particularly in the more distal ends of the tissue bed, as well as immune cells. And then the cells then, of course, make up the tissues make up the organs, and then organs together make up a more complete whole body physiology. And today, the focus is going to be on the tissue modeling efforts that we have going on here, and primarily emphasizing our efforts to bring in microvasculature into those tissue models. As a reminder, that microvasculature is an integral part of any tissue, or nearly any tissue. Cartilage, for example, is relatively avascular for many parts of it. It's necessary for tissue homeostasis, as well as tissue function. Of course, is serving as a conduit for blood and perfusion. That not only gives nutrient and gas exchange, but also inter-tissue and inter-organ communication, as signals and hormones, for example, are moving from one tissue bed to another. And of course, the vasculature serves as a gatekeeper there between the blood space and the tissue space, for example. Immune cells moving through the blood, moving into different specific tissue beds. And what we're learning also in our modeling, as well as others in their research efforts, is that the constituents, the cells of the tissue or the vasculature, communicate in a cooperative fashion with the cells of the tissue, whether it's the stroma or the parenchyma. And as we get further in, I'll give some examples of where we're seeing that in some of our tissue models. One other aspect I think that's important when it comes to tissue modeling, and it'll show up again in some of the models that I'll discuss here, is that the vasculature will take on tissue-specific phenotypes, so-called organotypic function, such that perhaps there's a continuous capillary bed in muscle and in brain, for example, where there's good segregation between blood and tissue space. Or as we move into endocrine organs, where we want good hormone exchange between the tissue and the blood space, there might be a fenestrated capillary bed. And then perhaps free exchange of plasma constituents with the tissue interstitium, such as you see in liver and with the sinusoidal. So always in building these tissue models, we pay attention to, is the vasculature taking on the characteristics of that specific tissue bed? And we think that's critical for that tissue function. It's also important to recognize that within the vasculature, we have multiple vessel types, not only in phenotypes, as I mentioned, with the continuous, discontinuous, fenestrated, sinusoidal, but also in the makeup of those vessels, and whether we're inflow, the arterial exchange, the capillary outflow, the venule, as well as all the different cell types, not only the different endothelium, of which can vary depending on where you are in the vascular tree, but also those cells that constitute the microvessel wall. And that cellular heterogeneity, we feel, is really important in allowing the vasculature to take on these organotypic features to adapt and remodel and fit the function of the tissue and matching then the vascular needs. And finally, no one single microvessel, say a capillary, really functions alone, unlike an artery where it operates as a single large conduit. If we take that artery out, for example, there's serious complications downstream. But if you take out a capillary, no real consequence, and that's because it's really the microvasculature exists as a network, a system of microvessel segments that are interconnected in a very hierarchical nature. And importantly, it can be self-assembled as well as adaptive as it's responding to those tissue cues. And we always like to think of the microvasculature as a cell system or a tissue unit of itself. So in reality, we're talking about the myocardium, where we have cardiomyocytes and starting to build muscle sheets and build that cardiac tissue. We actually have a microvascular tissue that's part of that. So really more of a tissue assembly when we think of these larger tissue beds that then contribute to the organ function. Our solution is to leverage what Mother Nature uses, and that is whole but fragmented pieces of individual microvessels. So unlike starting with individual vascular cells, which we did some years ago and had some success but struggled in other applications, we went to a different route where instead of going to single cells, we go down to the level of the whole microvessel and then work with those and enable those to create that new microvascular bed. We use adipose. In the left here is an image of that where the adipocytes, the fat-containing cells, are red. The vasculature is green. Strip away the adipocytes, leaving behind that microvascular network. Break that network up into small pieces, shown here in the middle panel. And importantly, those small pieces retain all the cells that normally reside in the microvascular wall, which further analysis has showed us that can be considerable. Not only do you have the endothelial cells and the smooth muscle and those perivascular kind of vessel wall cells, but we also have evidence of progenitor cells, stem cells, and including a significant proportion of tissue-resident macrophages. We think all of these are important in not only building a vasculature, but also in driving that tissue homeostasis, where the vasculature is contributing to really a moderation and a mediator of some of those activities that go on in that tissue bed. Importantly, if you take those microvessel segments and embed them in a three-dimensional tissue environment, here green collagen fibrils and then the red microvessels are growing. You'll see these new vessel segments that grow out via angiogenesis. They'll change direction and they'll actually find each other and connect and start to form this neovascular network. It's a dynamic system. It's a messy system. In the video playing here, perhaps you see a variety of different cell types that are moving around. Again, we think all of those are important to help start reconstituting as much of that tissue stroma as possible. Once grown up, if you then implant these, so if I start with my vessel isolates, I grow them up into a neovasculature and I implant them in a variety of positions. We've looked at peritoneal, kidney capsule, as well as sub-Q. We can derive a more stereotypical microcirculation. It was a good hierarchical organization, inflow-outflow paths, and all of this evolves from that initial neovasculature, and that really, I think, reflects that adaptation and plasticity of the system. And we let the tissue environment dictate the outcomes of the vessel and vasculature and microcirculation type. So in the process of doing these experiments, we uncovered this organotypic plasticity such that if we, in this particular case, which we published some years ago, take our fat microvessel pieces, combine them with cells that help establish the blood-brain barrier, a very tight permeability barrier in the brain. Those cell types are the astrocytes, and we use the precursor of the astrocyte, which we call GRP. If you combine those GRPs with our fat vessels in that 3D matrix environment that I just showed in the video, and plant those subcutaneously, it will evolve in forms of microcirculation that becomes more blood-brain barrier-like. So in the middle panel there on the right half, our fat vessels start to upregulate the GLUT1 transporter. It's a hallmark feature of the blood-brain barrier, and starts to show more restricted permeability than the fat vessels alone in that sub-Q position. And so we're starting to see this kind of parenchymal microvessel communication and cooperativity such that the vessels take on a tissue-specific phenotype. And I'll show you another example or two as we go on. With that in mind, then, I'd like to take you through some of the models that we've developed to explain a little bit how we're doing it, and then describing some of the biology that's going on, again, emphasizing the role of the vasculature in that. At Advanced Solutions, we're involved in building therapeutic tissues in addition to the tissue models, and then, of course, more streamlined assays for particular vascular-related functions. But in all cases, our strategy is to modulate the environment such that it enables the parenchyma and the vasculature to assemble and form a nice cohesive tissue environment. We build these tissue models and these therapeutic tissues using our bioassembly tissue fabrication platform. I'm just going to give one slide on this, but to highlight that this is our single robotic-based tissue fabrication platform where we have design software working and communicating with the robotic arm to do everything from bioprinting to tissue assemblies to pipetting, whatever is needed to build those tissues. And really take a design and make strategy with our second set of software called BioApps that allows this one single platform, which is agile and flexible, to build a variety of different tissue types. So with this platform and our isolated microvessel approach, this is where we really started to explore and develop those strategies and protocols for building these tissue models. And one such model is our thick vascularized human liver tissue model. We set out to really create a liver tissue environment such that we can look at cell migration, say Kupffer cell or immune cell dynamics moving through a tissue space, or perhaps house a hepatocellular tumor that might sit within a quote unquote normalized tissue environment. These are about a 1 centimeter thick. We use a bioprinting strategy to structure that tissue such that we get minimal necrosis and good media tissue exchange. And of course, we have our vasculature in there. We see good tissue function. I've shown here up to 7 days. We've seen this out to 2 weeks, 14 days. We're finding that having the microvessels present enables us to go to this thicker tissue environment and to scale it out to larger tissue volumes in general. There in the upper left is a 12-well example of our liver tissue. And interesting, where we're seeing our microvessels play a role is not so much in base functions such as urea production, but in cytochrome 450 upregulation, as well as biosalt transporter upregulation, etc. So really, our vessels seem to be adding to the refined functionality of the tissue, in addition to enabling this thicker tissue environment. Another example here is our vascularized islet models. We're really looking at it in 2 ways. One is, again, this thick tissue approach that we did with the liver, or in this case, using derived beta cells. We're working with a partner, Dr. Ipshita Banerjee at the University of Pittsburgh. She's deriving these beta cells from stem cells. And she's creating spheroids in her particular application. And we got together because she initially attempted to build a vasculature in these spheroids using isolated endothelial cells and struggled. And so we've been able to, working closely, devise a new strategy such as we can stage the fabrication of the spheroid and then get our microvessels into the spheroid, and then they undergo angiogenesis and really expand it. And we were pleasantly surprised to see that not only can we get that vasculature established, which, by the way, has implications if we want to start transplanting these, but the vessels themselves improve beta cell responsiveness to glucose, such as in a glucose challenge, we see this nice spike in insulin release, and then a return back to a baseline. Without the microvessels, the beta cells completely deplete their insulin stores and have to then be recharged. With the vessels, there's more of a balanced response, and one that you see more in the tissue bed. And interestingly, we start to see our fat vessels express more of that fenestrated endocrine-like organotypic vasculature that you see in the pancreas and in islets. And so this is our first example here of that cooperativity where our vessels are responding to the beta cells as well as the beta cells responding to the vessel presence. And this is a recent paper. It's in press. Just got it in the Journal of Biofabrication, where we're looking to build adipose organoids for use in adipose inflammation and insulin resistance studies, all related to type 2 diabetes. And here we're deriving the adipocytes from mesenchymal stem cells, and again, developed a strategy to integrate our vasculature. In the middle upper panel there, you see this nice red starburst of vessels that were formed in the spheroid. We place that spheroid into a collagen environment, again in vitro, and we have this nice robust angiogenic burst outside of that. So we know that our vessels not only are there, but they're functional. And again, this has implications if we want to tie this into other types of tissue models or perhaps pursue a therapeutic. Interestingly, while baseline adipocyte function, such as lipolysis, is good, but not really affected by the vessels that are present, the vessels in themselves induce the adipocytes to upregulate the insulin receptor, which obviously is important in not only glucose metabolism and fat function, but sets the stage for an insulin resistance modeling. So again, here we see this nice dynamic response of the adipose organoid to certain challenges, as well as some new features when we again start to reconstitute that tissue environment more with our microvasculature. In all cases, we've been exploring the utility of through tissue perfusion. In this particular example, it's with our thick liver construct. Using our bioassembly platform, we're 3D printing up sacrificial networks and assembling our thick tissue around those channels, and then using a customized system that works with off-the-shelf plastics such as multi-well plates that we've developed in-house. We can really start to provide perfusion through that tissue bed where the channels themselves are formed by the tissue. And so this gives us that added flexibility. We're certainly seeing changes that perfusion introduces, not only just in survival, but in some functional differences as well. And so I'll speak a little bit later about complexity, but our particular approach has been designed to give flexibility on how you assemble the model, depending on what types of questions you want to answer and what your objectives are. Finally, while originally I talked to you about how we can get our microvessels to form a neovasculature in vitro, you can take that and plant it in vivo, say under the skin of a immune-compromised mouse, and have that connect and inoculate to the host circulation and drive the formation of a new microcirculation. We wanted to recapitulate that full process in vitro. And so what I'm showing you here is our in vitro human microcirculation. We're getting ready to assemble this and put this in to review for publication. But we're combining our microvessel technology, brand named Angiomics, with a microfluidic channel system such that we have, and on the upper right perhaps is the best schematic of that, where we have microfluidic channels that we then grow adjacent a neovasculature from our isolated microvessels, similar to what I've been talking about so far, and inducing them to connect or inoculate with the channel walls that have been aligned with endothelial cells and form a network, and then start to, via intravascular perfusion and controlling hemodynamic cues, drive and mature that microcirculation. And so on the images here on the left, is upper left corner is our endothelial cell line channel. On the lower left is a neovasculature that's grown up, here shown in red fluorescence. In the middle are phase images where the two arrows, open arrows, highlight the walls of the channel, and adjacent to that is a network of microvessels that are formed. The arrows are pointing to some and certainly not all. The next adjacent panel is showing that inoculation event between are a microvessel from that network to that channel wall. On the right are stills from a video where we've introduced beads into the channels, and then we're observing those beads moving in through the different vessel elements of that microcirculation. And so like I mentioned, we're in the process of finalizing this body of work, putting it out there for review, and then getting input on what people think. Obviously, there's a lot of implications, both in therapeutics, but now also we're starting to think, how can we combine our other previous models, such as our adipocyte model, with the islet model, and start to really create a more complex physiological system where each of these model systems are connected via bona fide microcirculation, and really start to recapitulate as much of that in-body tissue environment and physiology as we possibly can. So with that, summarize a little bit what I've been talking about here with you. One is, the idea of recapitulating a tissue in vitro, recognizing full well that those tissues are complex and varied. Typically, when you start combining different components into a tissue-like environment, you start to see emergent behaviors. You start to see kind of a greater than the sum of the parts type behavior. And that's primarily due to that high integration and dynamics. So our modeling strategy is such that we enable those dynamics to play themselves out. Of course, we emphasize we have expertise in this. Then, of course, then a bias in the need to have a microvasculature in the tissue. We think it's a critical element. With or without perfusion, we're seeing evidence that the microvessels present contribute to good tissue model behavior. We also know that the vasculature participates in the disease process. We've not explored much of that yet. We're just starting to do that now that these models are in place. And we've gained the appreciation that they really serve the microvascular, and those elements of the microvasculature serve as a reservoir for stromal cells. In particular, we're paying attention to tissue-resident macrophages. We think that's key. And all of that really is starting to realize a microvessel parenchyma cooperativity that we just weren't seeing with endothelial cells alone. Some cases we were, but not in all. And so we're really pleased that we're able to advance this a bit further with our isolated microvessels. I went through a handful of models where we're building human vascularized tissues. One is that thick liver tissue model with and without flow-through perfusion. Modeling the islet there in the context of an islet organoid. This really where we have a good evidence of vessel parenchyma cooperativity. Of course, our modeling adipose inflammation and insulin resistance with our adipose organoid. And finally, we're really excited about our in vitro microcirculation model, which originally we set out to model tissue vascularization, but obviously we have an opportunity now to start to model a lot of other different biologies as well as physiologies. And with respect to strategies that we take to build these models, we don't always necessarily try to recapitulate a complete tissue, but those aspects of the tissue that we're most interested in. So perhaps it's cell-cell interaction, maybe it's dermal, subdermal dynamics that we want to see in the skin, or cellular composition. Experimentally want to reconstitute in different ratios or different cellular elements to understand the biology. Or compartmentalization, having a blood space that an immune cell can leave and enter into a tissue space, or perhaps a tumor cell leaving its primary tumor and then moving in and metastasizing out into a lymphatic space. All of that represents a lot of different dynamics that are relevant to health and disease, and even looking at biomechanics where we're altering tissue boundary conditions to see what the effects are on internal stress and strain dynamics. We're agnostic to the fabrication approach. Our fabrication bioassembly platform really is agile and lets us accommodate a number of different strategies, whether it's bioprinting, as I mentioned, or organoid formation that we then can assemble into larger constructs. But what's the biology you want to model? What's the best way to assemble that system, and how do you best implement that? And really, we walk through that kind of strategy routinely. Obviously, how complex you want to make it, how complex are you able to make it, certainly comes in and plays a key role. But ultimately, I think our goal is to start to assemble these multiple different tissue types to capture more and more of the whole body physiology. And with that, I very much appreciate your time, and would love to entertain and answer any of your questions. Thank you.